Asteraceae is a superstar rivaled by another. Okay. Great. Okay, and so because of this massive diversity, many botanists have been interested over the years in understanding evolutionary relationships within the family. And so one of the first treatments was by Cassini, and where he recognized 19 tribes, many of which we still recognize today. And next slide. And so with the advent of molecular phylogenetics, we also had a more clear picture of the evolutionary relationships in which we literally understood the family upside down. So learning that the early diverging lineages of Compositae are things like the Barnardizioideae, and things like Helianthidea are later derived species. All right, so from this work, many people have been interested in understanding using phylogenetics, molecular phylogenetics. So there's been work on the backbone, on the base tree by many colleagues. Many people have been interested in particular tribes, and so using many different markers across the world. And so in the Compositae book by Funk and colleagues, you can see there's a chapter for every tribe, so you can look at the phylogeny of those tribes. And then finally, understanding the supertree or the base tree using a meta-tree approach has been a goal of the group. And so what I'm showing here is a base tree of the family. So this is based on 900 genera and 10 chloroplast loci. And what you can see from this that I've circled and from the dashed lines, there are many regions of the backbone of the family that are sort of unknown. They're unresolved nodes to us. And so some of the reasons for this may be like many angiosperm plant families, polyploidy is quite common and rampant in the family. Great. And so when we had this question, we decided that we wanted to take a phylogenomics approach to this. And some of the reasons for this are we hoped that many loci would help resolve some of those regions that we were had in question. And also we could begin to tackle some of these questions that many of us here have been interested in understanding the differences between gene trees and species trees. And also next-gen sequencing has become affordable. And so as there are many techniques, one of the major techniques that people have used for phylogenomics is the Hive-seq. It's also called sequence capture or target capture or enrichment. And so there are many papers that have had methods that have either talked about the methods or that have used these methods, and our work has been one of those. Okay, and so I wanted to just sort of briefly tell you how we came up with our Hive-seq strategy. And so what we did was in about 2010, we took loci, about 1,300 loci that were single copy loci from Arabidopsis thaliana. And we looked for putative homologs in the sequence data of composites that we had at the time. So now we have sunflower genome and lettuce genome, but in 2010 we had ESTs, express sequence tags. So we had these for 3 species that were economically important: lettuce, safflower, and sunflower. And so we identified these putative homologs in our Compositae species, and from that, about 1,000 loci, we were able to say we can target these loci. And we call these the conserved orthologous sequences, or the COS loci. And so to design the probes, we worked with a company called, it's called Arbor Biosciences now, right? But it was called Microarray when they started. And we were actually one of the first groups to work with this company. So it was kind of neat to have them. They were a startup company, and now they're kind of a big company. And so we used this approach called MyBaits to design probes that would target these 1,000 genes. So I'm showing you here is maybe an example of just one locus. And I have these black lines here because, remember, we had EST sequence, so we had no knowledge of where introns were, how long they were, and that sort of thing. So we avoided intronic positions that we predicted based on Arabidopsis sequence. And so we tiled these probes that are specific to these loci across our genes, and then all the baits come in a tube, and you can order them from the company. Great. And so something that's really cool and exciting is that when we worked with the company, and maybe because we were sort of early on, we were able to have them make a lot of these probes, and they have them now commercially available. So if you are an Asteraceae person and you would like to try out the baits, they're available on their website to purchase, so you can buy the kit. And so I have the pricing up. This is from their website. And if you advance one more, so if you were to go and design custom probes from scratch, these are the prices on the right. For 16 reactions is $3,600. And so if you buy the kit, I sound like I'm selling it for them, but I kind of am, I guess. You know, it's much, much, much cheaper because they've made a large batch. So that's something that's really beneficial for studying the families, that we have this much cheaper price to be able to study phylogenetics. Okay. All right, and so just really briefly about taxon sampling, this has been something that we had to really think a lot about, almost more than the bioinformatics. And so we wanted to sample all subfamilies in the family, and so there are at least 13 subfamilies. We wanted to sample all the tribes, 40 plus tribes. We also wanted to make sure that if there were any monotypic groups, these sort of unusual, unique species, we wanted to make sure that we sampled those. We also attempted to sample diploids, but if everyone here knows, no plant is really a diploid. So we did our best on that. And then we tried to use the freshest material possible, but it turns out herbarium samples work very well. So we've used herbarium samples that are 30 years old and had no trouble with our methods. Okay, so just really briefly, because maybe if you were in the talk this morning, you saw a lot about next-gen sequencing, but the overview is that we make a DNA library. We usually do Illumina sequencing, but you could use, okay, thank you. I think I have a little extra time. Anyways, so I'm going to talk for longer than that. Okay, so you make a DNA library, and then you do this HiveSeq, the hybridization in the lab, and then you sequence using some sort of next-gen sequencing platform. Okay, so just really briefly, I won't run through this, but you make a genomic library, and we usually use a kit. So there are commercially available kits, or if you're very savvy in the lab, you can sort of make your own home brew, and you can get that for a lot cheaper. So we make genomic libraries, and what's great about that is, probably many of you know, but you can barcode, or you can give a unique tag to each sample, so that then when you sequence, you can pull all your species into one sequencing run, and then you can get a lot more sort of bang for your buck. So that's our method. Cool. And then in terms of the hybridization, it's an in-solution hybridization. So I've been telling a lot of people here that I think that it's easier than doing a DNA extraction. So nobody should be wary that their lab hands won't be good enough to do any of this work. So anybody here who can do DNA extraction can definitely do some HiveSeq. So this is just the method. So basically you do this hybridization in the lab, and then you can send out your samples for sequencing, either to your own university or to a company. All right, and so I just wanted to show this because the data that come out of the machine, it looks like Sanger sequencing data. So I mean, a little different, but the format of the files are in these FASTQ files, and then you can turn them into FASTA files, and so there is sequence data there. There's just a lot of it. So, all right, and so just kind of briefly about the bioinformatics workflow. So what we do is we have the sequencing, and I'm showing you maybe 3 species examples. So we have all this sequence data that we get, and what we do is to assemble each sample. We make a de novo assembly of the sequences. And that's the second step there. And then the third step is to then identify, we have all these longer contigs that we've assembled together. We need to know which COS locus they belong to. So there are various methods, things like Phylucci, HivePhylomaker, HivePiper, that you can use to assign orthology to your loci. So you match your contigs to particular loci, and then you make an alignment as you would with any Sanger sequencing, and then generate gene trees or species trees. All right. Okay, so now you get to see a tree. So this is a tree that we published last year. This is a pseudo-coalescent method, Astral. A lot of people have been using, and so this is 9 subfamilies that we published. We have about 25 tribes represented, 71 of the species, and we've color-coded based on subfamilies that were described by Funk and colleagues. And one of the things that I want to point out from this is that little tree on the side is one of the first trees that we published. And if you can see that the blue group there is non-monophyletic. So that is subfamily Chicorioideae. That's where things like lettuce and dandelion reside. And so early on in our work, with just a few samples, we found that that subfamily did not appear to be monophyletic with our nuclear data, although it remains so with chloroplast data. And so we thought this couldn't be, so we added more and more taxa. Thanks. I'm still going to go longer than that. So we added more taxa, and we still find that the subfamily is non-monophyletic. So we're finding lots of really interesting things using the nuclear data and using lots and lots of loci. And we have very good support for these new findings. All right. And I just want to point out, too, that one of the sort of features of doing next-gen sequencing is that you can often generate plastome data from the next-gen sequencing. So there's a lot of chloroplast DNA in your sample, and even though you are enriching for these nuclear loci, you often get the plastome, people call it bycatch or off-target sequences, so you can kind of get the plastome out. And then another thing that people are doing is if they think they may not get enough chloroplast DNA, then they'll add back in some of their genomic DNA library right before they sequence, so that they're sort of enriching for some more chloroplast DNA. And I want to note too that there may also be mitochondrial sequences that you can get from this. So that's a cool possibility. And then I'm just showing this tree to show you that we can get plastome data, and that the subfamily, for example, Chicorioideae, is still monophyletic with chloroplast. So that's something interesting, sort of biologically and evolutionarily to think about. Okay. Yeah, so just showing you sort of the difference. Yeah. All right. And so our current data set now comprises over 200 taxa, and we still find things like the subfamilies Cardioideae and Chicorioideae remain non-monophyletic with nuclear data, but monophyletic with plastome data, so it's, as I said, interesting. We have very high support for most all of the nodes across the tree. So it's really exciting that lots of data have generated lots of potential answers for the family. And what's really interesting is that if we compare our trees to lots of other composite trees, maybe that were constructed by transcriptome sequencing or chloroplast sequencing, in the composite, we see some changes, but we typically don't have these massive changes where we need to retell the entire story. So some plant families, different analyses may give a very different picture, but it seems that in composite, different methods, different analyses, we get a very similar general picture, and so that's kind of cool for us. And then one of the things I'll show you that we've been working on is to do some molecular clock dating of the family. And so we have fossils in Asteraceae. Most of them, except for one, are pollen fossils, and so there is one macrofossil, a capitulum from the Eocene. And so if we date the tree, I'm showing you now just some tribe level dates. And so we have an origin of the family that is Cretaceous. So I always like to tell my students, I want to know if Asteraceae was around when the dinosaurs were. And I think the answer is yes. And we're not alone. Two recent papers the trees that I'm showing on the left here, one by Jose Pinero, and then another one by Huang and Hong Ma and others, have also shown that there is a Cretaceous origin of the Asteraceae. So that's very interesting to us to think about, because you think about the origin at the Cretaceous, but then there was this mass extinction at 65 million years, and so it seems that that likely has had an effect on the family. So you can see that after the KPG, that's the line I have down, that there's more sort of diversification across the globe. And again, this is something that has been shown in some recent papers as well. Okay. So, of course, another question that we'll be working on is to understand then the evolution of the family, not just in time, but also in space. And so Vicky's work on biogeography of the family is very well known. And so we're hoping to add a biogeographic sort of aspect to our study to understand how this family came to be everywhere on the earth. And there are some hypotheses that her work has proposed. It's pretty much established that it's a southern South American origin of the family. The sister family, which is Cheliceraceae and only has about 50 species, is also South American. And so one of the big questions is kind of to understand how this moved in time and space across the globe. All right, so what have we learned? So we have all this data and we have all these trees. And so I'm showing you now again the image with the circles. And I think that in the black, I think we have a really good idea. So if you study these parts of the family, then you can talk to me. I think we have some good ideas about the relationships. But these that I'm showing in red, I think we still don't know the answers to those relationships. And so I think that more work in these red areas is certainly, more work in the whole family is certainly needed. And I think that it's very important for us to all sort of collaborate together. It's such a big family, and so just come talk to me. It's really exciting. Yeah, next slide. All right, great. So of course there's work underway. We're continuing to test these hypotheses about relationships in the family, and we have used this approach across the whole family, but what's really exciting is that it seems to work very well also at lower taxonomic levels. And so students in my lab and elsewhere have been working on particular genera or particular tribes, and we can all use the same markers, and so it's really exciting to integrate the data together. And so two of the talks today will be talking about using at lower taxonomic levels. And yeah, and so we're also interested in sort of understanding other drivers of diversification in the family. Polyploidy is one in particular. All right, so I think that's it. So I'll take some questions if you guys have any.